Well, good morning, ladies and gentlemen. Isn't it nice to be in Florida at this time of year, and especially in such a nice resort? I should like to thank the organisers of this meeting, NFSTC, and in particular Debbie Figarelli, as well as NIJ for inviting me to talk, or to be more precise, cajoled me to talk here on DNA mixture interpretation. And my task this morning is to basically talk about the history of DNA mixture interpretation. And this particular book that you see here is only available in limited editions. In fact, there's only one edition, and it belongs to me, and I could sign a copy later, but I don't actually have it with me at the moment. But as you can see, history of DNA mixtures is going to be an interesting topic, because what we're going to do is talk about showcasing ancient technology, and I mean ancient technology, meaning technology that's at least 30 years old. The science of mixture analysis, and then community consensus efforts. Okay, so the first thing that I'd like to talk about is, why are we interested in the history of anything? Well, obviously, those who cannot learn from history are doomed to repeat it. So we want to learn history because we don't want to make the mistakes of the past. However, we should also say, this is George Santayana, very, very famous saying. However, whilst we don't want to repeat the mistakes of the past, we don't want to be constrained by history either. We want to be able to be not constrained by history and move ahead with new technologies if we can get there. And hopefully during this meeting, you will be exposed to some of the newer technologies or newer approaches to mixture interpretation. So first and foremost, let's get the statistics jokes out on the table to begin with, because I'm not a statistician and I'm not a population geneticist. I regard myself as a forensic scientist, and therefore I'm quite capable of making statistics jokes, right? And I think the majority of people in this room, in fact, probably watching this also, are probably not population geneticists or statisticians. So let's talk about statisticians. A statistician is a professional who diligently collects facts and data and then carefully draws conclusions about them. That's one approach to thinking about what statisticians do. Secondly, you can lie with statistics, but you can even lie better without them. So you can lie with statistics, but they do constrain us in some ways also. And statistics, of course, means that you never have to say you're certain, I prefer the alternative phrase. Statistics means you never have to say you're wrong, which is a quite useful thing occasionally. But apart from these jokes, there are some seriously true precepts that we as forensic scientists, when applying statistical methods, should appreciate. First and foremost is that all models are wrong. So we use models in our approaches to mixture analysis, but only some are useful. Okay, and that was George Box, a statistician, who said that, and one of our colleagues in the forensic community, Bruce Heidebrecht, from the Maryland State Police, is very keen on using this particular phrase, and I am propagating it today. My favorite one, however, is from Nietzsche, the philosopher, and where he has stated there are no facts, only interpretations. So, and that's because we're human beings, and because the data we get come through our senses and through our central nervous system, and that central nervous system can be wrong, and it often is, but there are no facts. We interpret the data, and certainly we do that with mixture interpretation. And so mixture interpretation basically is a very simple concept in terms of what we're trying to achieve, and it's listed here, the procedure is listed here. First of all, we have to recognize mixture. Secondly, we have to infer the genotype. And third, we then attach a weight with statistics. So we need to recognize the mixture. That should be relatively straightforward. We'll talk about that, and so will other speakers. We have to infer the genotype or genotypes present in the mixture. And then if there's a non-exclusion or an inclusion, then we need to attach the weight with statistics. So the evidentiary weight of the match, if there's a match with a particular individual or individuals, then we need to attach the weight to it. So that's the statistics part, the third part. Now, to talk about the history of DNA mixtures, let us go back in time then to the pre-DNA era. And as you can see from this slide, this is an older forensic scientist. There aren't too many forensic scientists nowadays who wear suits and a hat, and not too many people use magnifying glasses either. So let's go back to way back to the beginning of forensic biology time, when we dealt with the ABO system. The ABO system, as you know, is a very important system for blood transfusion. But in the forensic arena, we did use it for many years because that's all we had, basically. So here's an example where a victim was typed, a female victim of a rape was typed, and she was found to be a B secretor. The vaginal swab with semen present was the evidence, and that evidence was typed and found to be a type AB. What that would mean, if there's a two-person mixture, is that the semen donor has to be an A or an AB. Okay? All bets are off if there's more than a two-person mixture, but if there's a two-person mixture, meaning you've got the victim secretions in combination with the suspect or the donor of the semen, then the semen donor must be an A or AB. Now, what does that mean? Well, in the population of Hong Kong Chinese, and I use this because I used to work in Hong Kong, and I'm using the Chinese population data here. This is from one of my old cases, that 27% were A, 6% were AB. Therefore, 33% of the Hong Kong Chinese male population cannot be excluded as donors of the semen stain. So in this case, 1/3 of all males in Hong Kong could have been the donor, but nevertheless, 2/3 of the males could be excluded. So that's ABO testing. We also had some other, with ABO testing, we used phenotype information, meaning that some of the genotypes could not be discerned because of dominance, etc. However, we also had some enzyme systems, one of which was PGM. And this PGM system had 4 alleles that were discernible, having 10 genotypes. And some of the genotypes are shown on the top there. Now, for a single genetic marker system, if all we have is a single genetic marker system, and often all we had was a PGM result, for example, then to recognize the mixture, you need to have a genetic marker with greater than or equal to 3 alleles, right? So there were some systems that were available at the time just to be able to recognize a mixture. We had PGM and we had other enzymes and serum proteins, EAP, GC, PGM, etc. But that's just to recognize the mixture. To be able to eliminate a proportion of the population as potential contributors, you need to have greater than or equal to 4 alleles. And there's only one system that did that at that time was PGM. However, if you have a multi-locus genetic system, and we didn't often have that, but if you did, then to recognize a mixture, you need at least one genetic marker with greater than 3 alleles. But to eliminate a proportion of the population, you need loci with at least one common allele that's missing from that. So you need an allele missing from the mixture. So, for example, if it's a 3 allele system, say it's EAP and there's ABC, if you have ABC alleles, that tells you, 1, that there's a mixture, but 2, it tells you nothing about the potential donors, because all individuals are potentially present in that. So you need to have an allelic system that allows you to discern when one of the common alleles is missing, as such. And we didn't have that very often in these days. So here's an example of the typical case that one did. This would be in the late 70s, early 80s. Here's on the left-hand upper left. Have I got a pointer? No. See if I can do it with my left hand. So here you have the 2 + 2 - 1 + 1 -. These are the 4 alleles present in the population. And say your evidence has a 2 + 2 - 1 -. So you can see that the evidence shows that there's a mixture. There's 3 alleles. And then what we would do would see, okay, what genotypes are included? So obviously 2 + 2 pluses, 2 - 2 - 1 - 1 - 1 - 2 + 2 minus, 2 + 1 - 2 - 1 - all the genotypes that don't have the 1+ allele present, which is missing. So all we would do, we'd go to a table of observed frequency tables of the genotypes. So here's the 1 plus, it occurs with a frequency of 35.7%, et cetera, You just add these frequencies up. We didn't care these days, that time, particularly about allele frequencies, apart from showing that by measuring allele frequency, by counting them and performing chi-square analysis, that the system was in Hardy-Weinberg, that type of thing. But in that era, we were talking mainly about genotypes. And so you just add them up and found, in this case, 16.2% of the population cannot be excluded or are included as potential donors of the stain. So, however, when you look more carefully at what that simply is, you're looking at the potential genotype combinations. When you look at the potential contributors here, 2 plus, 2 plus, 2 minus, 2 minus, 1 minus, 1 minus, when you look at the allele frequencies, of course, you can say that 2 plus is just P2 plus squared, 2 minus is P2 minus squared, 1 minus is P1 minus squared, 2 plus 2 minus is 2P2 plus, P2 minus, et cetera. So where the allele frequencies are P1 plus P2 plus, et cetera, that add up to 1. And then basically what you see is that in this particular case, if you just take the alleles that are present, P2 plus P2 minus plus P1 minus, and take the trinomial expansion of that, then you basically have the probability of inclusion, or the random man not excluded, which was, in this case, if you add the allele frequencies, gives 16%. The same as just by counting the genotypes, you'd expect that. And in these days, we were less than accurate, I believe, by just using the genotypes as opposed to allele frequencies in terms of the accuracy of the estimate. So we were used to, in forensic science, using the probability of inclusion, the probability of including someone, what would be termed the random man not excluded nowadays. That RMNE came from paternity testing nomenclature. So we were able to do that, do some mixtures in the pre-DNA era. But then, of course, DNA profiling came along, and now I'm going to talk about the pre-STR arena, because STRs brought us quantitative approaches to allele designations and allele interpretation, mixture interpretation. But if you looked at the older technology, quantitation was not used so much. So on the left-hand side, you have an RFLP gel showing, or autorad, showing, if it's an autorad, showing clearly here a mixture of DNA in this line here, for example. Why? Because there are 4 bands, at least. In that, there are 4 bands there. So it's easy to determine a mixture. Is that pointing? Oh. Inside? It's a pen. Oh, okay. Okay. I need to use that. So we have obviously an obvious mixture here. That's RFLP. And here's, and how many people have done RFLPs in here? Well, a lot of people, good. And then we have the dot blot, reverse dot blot system, DQ alpha. How many people have done DQ alpha here? You have. Okay. So you can clearly see that with these older systems, it's easy to discern the mixture, right? And there were methods to try and deconvolute that mixture. And most of them were based upon, certainly in the United States, random man not excluded approaches. And of course, this case that is quite a famous case, that a good percentage of the people here were born before this. Some may not have been. But this is the O.J. Simpson case. And in the O.J. Simpson case, the evidence of the O.J. Simpson case, there was a crime scene, Bundy, then there was a car in which the suspect was supposed to have left the scene at, and then there was a residence where the suspect lived. And you can see that for a lot of these stains that were found, there was a mixture. There was mixtures found. And in fact, mixture calculations were used in this case. So that's 1995. So this is the pre-STR arena. Okay, so that's a preamble then to now, I want to now talk about the STR kind of arena. But let me, before I do that, talk about the science of mixtures. And Dennis Lindley, who's a very famous statistician that has and Dennis Lindley's work has influenced a lot of statisticians, especially from the Bayesian standpoint. He's quoted as saying, it is only the manipulation of uncertainty that interests us. We are not concerned with the matter that is uncertain, thus we do not study the mechanism of rain, only whether it will rain. Now, that's not how we think, I think, as forensic scientists, but it's interesting to see how the statistician approaches the problem. So we have to be aware of the fact that we approach the problems often from slightly different angles, but hopefully we can bring the two together. The empirical studies that we do, we want to know the mechanism as well as the statistics at the end. So let me talk about the early evangelists for the science of mixtures. And I should make an apology right at this moment to say that this is not supposed to be an academic treatise. I'm going to miss people out. I'm going to cause problems with some individuals who aren't quoted, etc. I'm sorry about that. I've only got a few minutes, and I can't do it. It's not an academic treatise. This is setting the scene for a mixture interpretation workshop. But I'll try my best. This is my own personal view as to what's happened. So there were early evangelists, and in terms of the science of mixtures, and primary amongst them is a gentleman by the name of Ian Evatt, who worked at the Home Office Forensic Science Service, and some people here know Ian Evatt very well. And he published a series of papers in the 80s that, again, there's work before the 80s, but I'm referring to Ian Evatt's papers now in terms of the forensic science aspect. And so he published a paper in 1983 saying, what is the probability that this blood came from that person, right? And remember, this is pre-DNA era, right? What is the probability that this blood came from a person based upon standard genotype analysis at that time, which would be ABO and enzymes? And he introduced the framework, the likelihood ratio framework, instead of coincidence probability. Coincidence probability meaning, okay, that the 16% of the population, have this particular blood group or this enzyme group, or 16% cannot be excluded as components to this mixture. That's coincidence kind of probability. He introduced that as a logical framework, but this was on the basis of a single source, a so-called single source sample. And I distinctly remember myself having arguments with Ian Evatt, not arguments, discussions, about what's the point of this, because the coincidence probability and the likelihood ratio are the same, right? Now that was early on. I'm talking about the single sources, right? And we'd have many, many discussions in bars that shall remain nameless and things of that nature. But he was a true evangelist. And so many of us were very skeptical about okay, it's nice. This is a statistician's approach. So what? It's interesting. Coincidence probabilities, it's the same result anyway. That's what we thought. And I remember Ian Evatt himself. Ian Evatt was, if I remember, was a document examiner, a forensic scientist, document examiner, who decided, I'm going to go back to school. And he went back to school, and it became dangerous, because I think he studied mathematical statistics or something. Some of the gentlemen over there may know more. But he went back to school and learned some mathematical statistics, and came back into the forensic science service and really revolutionized it. I would call it revolutionized it now in terms of the thinking. So it took him a few years to be able to get his arguments together, I think, because in 1987 he published a paper on meaningful questions, a two-trace transfer problem. Now you think a two-trace transfer problem, what do you think that's going to be about? It was DNA. So we now talk about trace DNA. He used that term a long time ago. It wasn't DNA, it was trace, a question of trace, because biological evidence is trace evidence. So I wanted to come back to this paper, because this is a much more meaningful paper in terms of it maybe started to persuade people, such as myself and others, that maybe he was onto something. Our initial skepticism was changed by this paper. And then I'm going to come back to a couple of these papers. In 1991, he published a paper on that 87 paper was still on conventional grouping. In 1991, a guide to interpreting single locus profiles of DNA mixtures. This is now him applying the likelihood ratio approach to DNA mixtures. And then in 1998, when the STR arena was in vogue, you can take into account peak areas when you're doing this mixture interpretation using a likelihood ratio format. So that two-trace transfer problem paper, he uses conventional markers and phenotypes, frequencies, what we would call genotype frequencies now. He would call it phenotype because of the possibility of ABO, I believe. But anyway, think of example one. He gives two examples. Example one is just a single donor. A crime has been committed by a man who left a blood stain at the scene. The blood is typed using polymorphic genetic marker systems that have a number of distinct phenotypes, genotypes, gamma 1, gamma 2, gamma 3, different genotypes. So in the PGM system, there would be 10 genotypes, G1, G2, up to G10. And they occur in the general population with frequencies Q1, Q2, Q3. These are not allele frequencies, these are genotype frequencies. So the scientist has evidence. The blood stain is typed as F, the evidence is F, and the blood stain is typed as gamma 1, so a particular genotype. Remember, this is a single source. The suspect is typed as gamma 1, so the stain and the suspect are the same genotype. So the two hypotheses are C, that the suspect committed the crime, and or not C, the suspect did not commit the crime. Now, nowadays, of course, we wouldn't use that. It's not whether the suspect committed the crime or not, it's whether or not the suspect left the evidence as opposed to committed the crime. But that's okay. So in example two now, because we're going to compare the two, the crime has been committed by two men who both left a bloodstain at the scene this time instead of one. And the scientist has evidence F. The bloodstain is typed as gamma 1 and gamma 2, so there was two genotypes that can be discerned. And the suspect is one of these genotypes, gamma 1. And again, C is the proposition that the suspect was one of the two men who committed the crime. One of the two men who committed the crime was the suspect. And C bar, or not C, is the suspect was not one of the two men. It wasn't one of the two men who committed the crime. So the likelihood ratio then, as he formulated, Ian Evett, is the probability of F, given that C is true, versus over the probability of F, given that not C is true, that the person did not deposit, or wasn't guilty in this case. So the likelihood ratio of example one is simply 1 over Q1. 1, because what's the probability of the person depositing this? If it's only a single donor and it was his genotype, then it's 1 over the frequency Q1, which is just the frequency that someone at random could have deposited that. It's just the frequency of that genotype in the population. However, with example 2, we have to take into account the fact you have two donors. And so what's the probability of the suspect and someone else leaving that stain? It's 1, because the suspect is 1, times the frequency of that the other donor's genotype, over the possibility that, what's the probability that two random individuals would have deposited this? Just the frequency of one genotype versus the other, but because male 1 can have Q1 and male 2 Q2 and vice versa, you multiply by 2. So likelihood ratio in the first example is 1 over Q1. The likelihood ratio of the second example is 1 over 2 Q1. So this is the interesting thing then, you see. So suddenly then the likelihood ratio is a half that of the single stain. So the evidence is less probative. So now we're saying, wait a minute, the coincidence probability is not identical in this situation. Now you could argue this. You could argue the same logic can be used, of course. But interestingly, if you have n different blood stains, if you now have, instead of 2 donors, you have n different blood stains of different types that can be discerned in that group, then the likelihood ratio is 1 over n Q1. That is the reduction in the likelihood ratio. It's been reduced, right, each time by the reduction in LR is dependent upon the number of donors and not upon the frequencies. So if there's 5 donors, the likelihood ratio is reduced by a fifth. Okay? So this tells us that the degree of probability of the evidence is going to be decreased by the number of donors under this scenario. And also, if Q is greater than 0.5, the frequency of Q1 in this case, then the likelihood ratio will be less than 1. It can be less than 1. So the evidence reduces support for guilt versus innocence, so to speak, in this. Now, this is a totally different concept to the concept of just a simple coincidence probability. And this paper and subsequent papers from this, and it takes years for it to diffuse into the community, has had major impact on how we think about evidence and particularly mixtures now. So then his next paper was that I referred to was interpreting single locus profiles of DNA mixtures. This time, instead of using genotype frequencies, he was using DNA markers this time instead of traditional markers, and using band frequencies or allele frequencies. And here, he was able to show that if you take an example 1, a 4-allele mixture, ABCD with a suspect possessing 2 of them, you can get a likelihood ratio, and you can calculate the likelihood ratio. We don't have time to go into it, but at the moment, it's 1 over 12 FBFC, right? That's if there's a suspect possessing 2 of the 4 alleles with an unknown suspect. If a second suspect is arrested and he ties in with the evidence and has A and B alleles, so that all alleles now are accounted for in that proposition, then the likelihood ratio is correspondingly improved, because it's instead of 1 over 12 FBFC, it's 1 over 24 FBFCFD. So it gives another example of, that first example is when there's 4 alleles and they're all accounted for. There's 4 alleles and therefore it's a 2-person mixture, so therefore there's certain limitations. Example 2 was when there was a 3-allele mixture and you have ABC with suspect possessing 2 of them. The likelihood ratio equation becomes a bit more complicated, and the likelihood ratio equation will become more complicated if you then have to take into account dropout in an RFLP gel, etc. It becomes more complicated. So as he quite rightly states, as the number of alleles increases, the LR evaluation becomes quite complicated. Right? So, and the evidential strength falls rapidly with increasing numbers of alleles in that mixture. These are all facts. No, they're only interpretations. Okay. So then he moved on to, so that was based upon RFLP, where you're taking just the presence or absence of alleles bands. However, to take full advantage of the STR-type systems, we want to take into account peak areas when interpreting mixed profiles, so he published a paper on that. This is a conceptual paper that establishes logical framework for taking into account peak areas. Right? Because once you start taking peak areas, you have quantitative information, you can then decide that certain potential mixtures can't be present because of the ratio of the peak heights, or peak areas in this case. And the use of peak area data and mixing ratios permits the ranking of different likelihood ratios that individually evaluate all possible combinations of genotypes present in the mixture. And we need computer programs. So that was Ian Evans' contribution, major contribution. And he's made more than that, but that's what he's made in terms of this talk. Then we come across an interesting paper an important paper from the Forensic Science Service. Notice the Forensic Science Service has played a major role in this, by Clayton and Gill and Whitaker and Sparks, but they're known as the Clayton paper. The analysis and interpretation of mixed forensic stains using DNA STR profiling. And this is an important paper because this is what we basically do nowadays. You identify the presence of a mixture, you identify the number of contributors, or postulate the number of contributors, determine the approximate ratio of components in the mixture, determine the possible pairwise combinations for components of the mixture, and compare the resultant profiles for the possible components with those from the reference sample. And step 1, to identify the presence of a mixture, extra bands, allele peak symmetry, straightforward. So we can detect the presence of a mixture quite easily. Identify the number of contributors, it's a bit more difficult, but obviously The maximum number of alleles at a locus is 4 for two-person mixture, 5 or 6 alleles indicative of 3 or more. Experience indicates the majority of mixtures encountered in casework are two-person mixtures. That's wishful thinking, obviously, especially with present touch DNA. But I think the data, we have some data that Swigdam put together that do suggest, as far as we know, two-person mixtures are much more common than 3 or 4 or more. Determine the approximate ratio of the mixture, components of the mixture. The weight ratio is important. And of course, if you use the Mellogenin system, that's a good system that one can use to apportion the weight, the ratio of the components of the mixture. But of course, one can use other loci as well, of course. But this paper suggested the use of Mellogenin because it's relatively straightforward. Of course, there's some caveats to this. Of course, you have mutations, etc., etc. XY. In some individuals, XY is not equal. Does not have an equal peak height because of some issue, some genetic, some biological issue. Anyway, so you determine the weight ratio, and as I said, you can use a Mellogenin and others, but this paper used Mellogenin. Determine the possible pairwise combinations. If you have 4 alleles, there are certain combinations. There's six combinations if it's a two-person mixture. 3 alleles, they're listed in the center here. 3 alleles. If there's 2 alleles, you have certain. So there's only certain combinations that are possible. And so using the quantitative information drawn from the peak areas in the profile and the approximate ratio of the mixture, some of these pairwise possibilities can be discounted. So these are the list of possible pairwise contributors, but these can be some of them can be eliminated because of quantitative information. And then you compare the resultant profiles with those from the reference sample. Okay. Later evangelism, so that's the Clayton paper, which is a framework that I'd say most people, or a large number of people, would use and believe is the best paradigm at the moment for how you do a mixture case. Now, I would call this later evangelism, and I would refer to Bruce Weir, Department of Statistics, North Carolina State, now University of Washington, and there are several papers that have emanated from Bruce's group, as well as John Buckleton, who's we're lucky to have with us today. 1997, there's a paper interpreting DNA mixtures, and that's an important paper because that refines and expands the likelihood ratio concept. But more importantly, for many who work in the area, it actually provides how-to formulations. There's a bunch of formulations in there, and the basis of what these formulations, how these formulations were calculated for the most part. And so you have a long list of the LR calculations for various scenarios, because it becomes quite complicated, if you remember. And so that's an important paper from that perspective. 1999, 2 years later, using the same framework, or same approach, using a likelihood ratio, this is Curran, Triggs, and John Buckleton and Weir. Things get a bit complicated now because, maybe we need to take into account population substructure. Now we have to not only deal with statistics, now we have to deal with population genetics as well, which is life, right? So that's what we have to do. So we have to take into account, or we can take into account, effects of population substructure, the role of evolution in shaping the probabilities of sets of profiles, because not every profile is possible in this world, and some particular profiles are more common in certain populations than others because of evolutionary relatedness, etc. And it counts for the information. If you use these formulations with the appropriate subpopulation substructure formula, then you can account for information contained in the profiles of people who are declared not to have contributed to the evidence profile. John can explain that what that means later. Now we have so that's the modern evangelists, so to speak. There's postmodern evangelists as well now, and I think John Buckleton is in both. He's a modern evangelist and a postmodern evangelist, I believe, but he can speak for himself about that. With the postmodern group, we have to take into account PCR artifacts. So, for example, 1998, interpretation of simple mixtures of when artifacts such as stutters are present, with special reference to multiplex STRs used by the Forensic Science Service, Gill, Sparks, and Buckleton. So not only do we have to deal with statistics, likelihood ratios, number of who's in the mixture, the proportions, population substructure, but now we have these artifacts caused by our scientific process. We have to deal with them as well. And so these artifacts are stutter, next paper, 2009, interpreting low template DNA profiles. Doing DNA profiles, low template DNA has certain characteristics and technical artifacts that we have to deal with if we're going to interpret mixtures. 2010, a universal strategy to interpret DNA profiles that does not require definition of low copy number. So we're into this phase because of touch DNA, etc. We have to have some means by which we can deal with low template DNA. Anyway, so that's a likelihood ratio approach. Meanwhile, back at the random man not excluded ranch, or the probability of exclusion or inclusion. There's a paper that's always quoted, a 1993 paper by Devlin, and this is supposed to tell you how to calculate the probability of exclusion or probability of inclusion. I couldn't find it anywhere, but that's what it's always quoted as being the paper that refers to how you do probability of exclusion and inclusion. My belief is that these terms, probability of exclusion, random man not excluded, they came from paternity, the paternity side of the house. But things were but many people did use the random man not excluded approach. As I said, with PGM, with ABO, that's what we did, right? But there was a kind of not a lot in the literature until this paper, 2009, by Bedoli, etc., where this was looking at mixtures where, of course, we should use a stochastic threshold. So the formalized use of a stochastic threshold. And this paper, I don't have time to go into that paper, but of course I'm sure many people in the audience are familiar with the paper, where one needs to use a stochastic threshold to discern whether a peak is on its own or has a hidden daughter somewhere or not. Okay, so is an apparent homozygous really a homozygous, or is it a heterozygous contributor type of thing? But in this paper, it's quite clear that the authors state that a standardized mixture interpretation protocol is not recommended or possible, right? So, and the authors clearly prefer the random match probability and probability of inclusion approach instead of a likelihood ratio, because the random match probability of inclusion conveys to the trier of fact the probative value of the evidence in a straightforward fashion. And so I think what we're going to hear in this next few days is a discussion on this approach, the approach of random man not excluded, as well as the likelihood ratio. So hopefully participants will be exposed to, you will be exposed to both approaches in this workshop. Now there are current evangelists that now talk about quantitative data modeling. I'm Mark Perlin, for example, of cybergenetics is one such individual who's one of these current evangelists. And these types of models, there are possibility of using hierarchical Bayesian models with Monte Carlo, Markov chain Monte Carlo solutions is possible. So this appears to be a standard approach in modern science, certainly, but not forensic science. It describes uncertainty using probability. It's been described as a new calculus. It replaces hard calculus with an easy computing, can solve virtually any problem, and is well suited to interpreting DNA evidence. Now, this is from Mark Perlin. This is Mark Perlin's slide. So, but there's no question that from what I can see, the field is moving towards considering these hierarchical Bayesian models or quantitative modeling. So this diagram is from James Curran's paper, MCMC, Markov chain Monte Carlo, method for resolving two-person mixtures. And basically, this is a graphical model. And basically, the bottom line is you get data variation. It's caused by the genotype, the weight ratio, and the peak areas, etc. And there's parameters that affect all of these. And this can be done. This it's a simulation. It's done via computation. You require computation. And before we move on to a model like this, we would need to be retrained. When I say we, the forensic community, most people in the forensic community have done statistics 1. That's descriptive statistics. Normal distribution, binomial distribution, test of hypothesis testing, regression. Some may have done statistics 2 with analysis of variance, non-parametric statistics. How many have done statistics 3? Which does it even exist? Statistics 3 are all these modern methods. When you read papers, everyone does these methods. Well, everyone does them, but nobody understands them, because in our community anyway, because most of us have not taken courses or have had access to individuals who could train you in these modern methods. But please believe me, they're with us. Are there software solutions for mixture deconvolution? Yes and no. There are the theoretical basis for software solutions are there. Linear mixture analysis, least squares deconvolution. There's the pendulum system that Chris McGuire could talk about till he's blue in the face. But I would say that, which is part of the FSSI 3 software suite, still, yes. But I should say that at New York City OCME, they've developed a program recently that uses likelihood ratios with probability of drop in and drop out. And that's an interesting approach that they're taking. And apparently that may be made available to the community for those interested. And the mixed DNA profile then, you can have, this kind of summarizes some of the approaches. The frequentist approach, this is from John Buckleton's book, which was originally from Tim Clayton and John Buckleton, Chapter 7, Mixtures, John Buckleton's book. So the frequentist approach, you can have the frequentist approach, which is exclusion probability, random and excluded, or you can have Bayesian approaches. Qualitative data, which is the likelihood ratio approach of Ian Evett and others, and which we are early on, or we can have more quantitative, early LR, or we can have quantitative and qualitative data approaches, where you can have the binary model, where there's quantitative information used to discern, to produce likelihood ratios of differing strengths for the same evidence, or you can have this continuous Monte Carlo model with Markov chain Monte Carlo simulations, etc. So that's where we are at the moment. So, with 2 minutes to go, or 3 minutes to go, I want to talk about the community efforts in this area. So we had, meaning that's the science of mixtures. Now I want to talk about what the community has done, like forensic science by fiat, forensic science by committee. Because a lot of people in this business, as we know, like to have the security blanket of a SWGDAM or TWGDAM guideline or some other guideline, ISFG guideline, so they can have that security blanket when they go to court, right? So what has the community done? Technical Working Group on DNA Analysis Methods, 1989 and 1991 and 1995 produced guidelines, the TWGDAM guidelines. And 1991, what did they say about mixtures? Validation, mixed specimen studies should be carried out, investigate the ability to detect the components of the mixture. That's all. Analytical procedures, when semen is identified, a method of differential extraction should be used, and when appropriate, each of the DNA fractions typed. So there was a requirement when there was a mixture, when it's a sexual assault, to try and separate that mixture. Why? Because you don't have to deal with the mixture to begin with. So why did we require this differential? So we could not deal with mixtures. That was the whole point. So, but it's basically silent. So basically, TWGDAM 1991, TWGDAM in 1995 said the same thing. That's my time. Stop. And basically, they were basically just determine the presence of a mixture. That's basically it, or do a differential extraction. The NRC, National Research Council, in 1992, says if a suspect's pattern is found within a mixed pattern, the appropriate frequency to assign such a match is the sum of the frequencies of all the genotypes that are contained within the mixed pattern. That's the random man not excluded. So that was what the NRC said in 1992. 1996, they changed their mind a little bit. And referring to the previous 1992 calculation, this calculation is hard to justify because it does not make use of some of the information available, namely the genotype of the suspect. The correct procedure, we believe, was described by Evett et al. 1991, that you now know about, likelihood ratio. That was 1996. Then we had the DNA Advisory Board. And that's some of the DNA Advisory Board. And there's at least one Nobel Prize winner in that picture, if you can find him or her. The DNA Advisory Board was set up by the DNA Identification Act 1994, set up the QS standards, and basically no substantive changes to the Twigdam guidelines for mixtures, except it said validation, species specificity, sensitivity, stability, and mixture studies are conducted. Okay, so you conduct mixture studies, whatever that means. Analytical procedures, the lab shall have a procedure for differential extraction. So the DAB, and I was part of that DAB, we said nothing about the interpretation of mixtures, other than to do studies on it. That was the early DAB, the first DAB. However, in this publication, 2000, this second iteration of the DNA Advisory Board talked about statistical and population genetic issues affecting the evaluation of frequency of occurrence of DNA profiles. And in this, they talked about mixtures. DNA samples derived from 2 or more contributors, presence of 3 or more peaks, elucidation of a contributor profile straightforward sometimes. When intensity differences are sufficient, it can be treated statistically as a single source, so you can take major, minor. It may be difficult to be confident regarding the number of contributors, but you can infer the number by reviewing data at all loci. When you can discern the contributors of a DNA mixture, there are 2 calculations that convey the probative value, the probability of exclusion, and the likelihood ratio. So the DNA Advisory Board recognized that there's 2 calculations now, RMNE, despite the NRC 2 saying don't do it, we don't like RMNE, the DNA Advisory Board came out and said you can do 2, LR or RMNE. And of course, then we had the July 2000 STR interpretation guidelines, because of course we have today's one, but I'm not talking about the 2001. And in this, the interpretation of results, there's a bit more in these guidelines, not much more, but certainly you can distinguish different types of class of DNA profiles. Single contributor versus mixture with major, minor, versus mixtures with a known contributor, like an intimate sample type of thing. You can then subtract the victim in an intimate sample, for example, or mixtures with indistinguishable contributors. So at least we're beginning to categorize these. And in terms of the statistics, however, remember that's recognizing the mixture and trying to infer the genotype. In terms of calculating the statistics, the formula used in calculating frequency should be defined for the following. So that's what it tells you. That's what to do. But how do we perform the calculations? Silence is golden. No guideline whatsoever as to how to calculate on that using the Twigdam interpretation guidelines. Just know that you can now infer genotypes, but then when you get the genotypes, we don't know how to actually calculate this, or we haven't told you how to do it. Of course, the ISFG, meanwhile in Europe now, the ISFG Recommendations and Interpretation of Mixtures, and here are the authors, John Buckleton again is part of that group, and they basically, I'm not going to get into details because of the time, but basically likelihood ratio is the preferred method according to ISFG. Scientists should be trained in use of likelihood ratio. Methods to calculate likelihood ratios are cited. Follow the Clayton approach, the paper I showed you about, the Clayton paper. Prosecution determines the prosecution hypothesis, defense determines the defense hypothesis. When minor alleles are the same size as stutters, then they're indistinguishable. Allele dropout to explain evidence can only be used with low signal data. You can't just say, oh, it's dropped out unless there's some evidence that dropout could occur. No statistical interpretation should be formed on alleles below threshold. That's a bit controversial. And stochastic effects limit usefulness of heterozygote balance in mixtures. And but these recommendations are recommendations only. And Debbie, I'm going to finish. These recommendations have been written to serve 2 purposes: to define a generally acceptable mathematical approach for typical mixture scenarios, to address open questions where practical and generally accepted solutions do not exist. This has been done to stimulate the discussion among scientists in this field. The aim is to invite proposals and criticism in the form of comments and letters to the editors of this journal. We're hoping to continue this process. See, sometimes scientists put something out there to stimulate discussion. It's not the be-all and end-all. So sometimes you go to court and say, this is the document. Well, if you read carefully that document, you have to be careful. There's some things in that document you may not want to hang your hat on. It's a discussion document, basically. And there are various responses to this document. The UK response, the German State Commission, the MC policy statement, the Australia-New Zealand support statement, most of them accepting the fact that likelihood ratios are the appropriate way to do things, but with the proviso that you can use under certain circumstances, RMNE, and if you have to, some more for the RMNE approach, some less so. NIST has done a lot of good work in this in the United States by showing that by sending the same electropherograms to various labs throughout the country, that they come up with different with the same electropherograms, same allele frequencies, same data. It's the interpretation is different. And so you can have the same sample having a likelihood or a frequency of occurrence anywhere from 10 to the 15 to 400,000, right? So that's a big 10 to the 5 to 10 to the 15, 10 to the 10 fold difference in the same data, right? So there's a certain issue there of standardization. Now, in July 2009, there was revised quality assurance standards issued through SWGDAM, and then developed by SWGDAM and then promulgated by the FBI director. And in terms of mixture, here's the only things we can find in that document, and this is 2009. A casework CODIS administrator needs to have documented mixture interpretation training. Internal validation studies should be conducted that require mixture studies. Internal validation shall define quality assurance parameters and interpretation guidelines, including guidelines for mixture interpretation, whatever they are. And labs analyzing forensic samples shall have and follow a documented procedure for mixture interpretation that addresses major and minor, inclusions, exclusions, and policies for the reporting of results and statistics. Again, silent on how you actually do this work. Now we're at the present, so-called, and we have the SWGDAM interpretation guidelines for autosomal STR typing by forensic DNA testing labs, and we're going to hear a lot about that in the next sessions, I believe. So that's me trying to bring you up to date with my perception anyway as to where we are with mixture analysis and where we might be going in the future. And sorry for taking a few minutes extra time. Thank you.